taking a look at Unit 3, Lecture 4, Innate Immunity. In our last lecture, we talked about the overview of the immune system, and here we get really into the first and second levels of defense, which are both considered innate immunity. So, the innate immunity, as I just said, includes both the first and second lines of quote-unquote defense. And what lies after that is the more long-term, slow-moving, acquired defense, as you can see on this chart here. So the idea of the first level and the second level together being the innate immunity is that they end up providing more of a rapid but non-specific kind of response to infection. And as we'll look at in the next chapter, the third level of defense, the acquired defense, it takes longer to develop, but it also lasts longer, and it's highly specific to exactly what you're being infected by. And as we'll see here today, that the innate response is generic to things that our body simply perceives as foreign. So there's sort of an innate concept of foreignness that supersedes individual species. Now, ultimately, what this level of defense does is it prevents pathogens from establishing, or at least it attempts to. So if you go back to chapter 13, establishment is the idea that they have to enter, attach, and survive the host immune response. So this is now the idea of what it is essentially they're trying to survive from. Now we'll start off with some different concepts, but that's what we'll kind of end up talking about in the majority of this lecture. So key concepts, we start off with physical barriers, and those largely consist of skin and mucous membranes. However, we also have our own microbiome, the natural resident microbiota, and a certain level of chemical barriers. Both of those together really consist of the first line of defense. Then we get into a very key part of the second line of defense, which are phagocytes. These are cells that eat other cells. Then we get into inflammation, which can lead to fever in extreme cases. And then we have another line that consists sort of broadly here of antimicrobial products. So we'll talk about two of the most well-known of those antimicrobial products. So all of that really together summarizes in a broad sense all of the different aspects of innate immunity. And so when you go back to the idea that microbes have a difficult time of infecting the body, these are all just some of the early barriers that they have to get around. Got to get through the skin, get past the mucus, get around the natural biota, avoid chemical barriers, avoid being eaten, avoid the effects of inflammation, and avoid the effects of antimicrobial products. This is why it's very difficult for pathogens to actually establish in the body. And a lot of that comes down to either your body not being able to keep these things maintained very well, or them finding very specific areas of the body where they are not existent, or primary pathogens having specific adaptations that allow them to get around these things. Now, the physical barriers come down largely to the skin being the big one, and specifically the outer layer of the skin called the stratum corneum. So this figure here is not in your textbook, more of an anatomy physiology figure, but the skin consists of multiple layers, and we're not going to get into all of that. But as you get to the outer layer of the skin, what you'll find is this dead skin cell layer. The outer layer of your skin is actually dead. So you can't really kill or harm something that's dead, and so your body very strategically surrounds itself with this dead layer that separates the inner and the outer compartments of the body, so to speak. Now, this layer is constantly, in addition to being dead, it's constantly being sloughed off, as they say, through desquamation. Sloughed off just means that the cells are kind of constantly falling off, and the way it works is that new cells are constantly growing up from the lower levels and pushing those cells off. And so as a new growth emerges from the bottom, the old cells fall off, and there's a constant replacement. The living cells die and become the replacements of the dead cells at the top. That's an ongoing, constant process throughout your entire body. So ultimately, it keeps a dead layer on the outside, and it constantly pushes those layers off so that bacteria can't really ever stay there permanently. Now, that's on the outside of your body. On the inside of your body, you have a lining of mucus, which I'll show in this next slide here with the picture, that lines largely the digestive, urinary, and respiratory tracts. You also have this around your eyes, the openings in your eyes as well. But what this ultimately does is it acts as a sticky kind of trap, if you will, that makes it very difficult for microbes to get through it and very difficult to attach to it. And it's basically like a sticky trap kind of all around the internal parts of your body. So the skin is kind of the physical barrier for the outside, and the mucus is largely a physical barrier for the inside. This figure here shows what that looks like. The inner parts of your body are made up of epithelial cells. These are cells that line the exterior along the inside of your body. So in other words, these are kind of open to a cavity in the body. So microbes can come in to different portals of entry into these cavities, but before they can get to the living cells here, they first have to go through a mucus coat and a layering of mucus here. So as long as that's relatively healthy and constantly being produced in sufficient amounts, that makes it very difficult for microbes to penetrate through that to get to the living tissue down below there. And then even when they do that, as you can see here, they do penetrate through that, there are other cells that are activated in that area that eventually come and try to remove them once they make it past there. And so that's kind of the idea. The physical barriers are not going to be foolproof. Things will get through them. But they are largely inhibitory to most things, and those that do will be dealt with in a different manner. So in addition to the skin and the mucous membranes, a couple other aspects of the physical barriers include your own microbiome. They exhibit what's called microbial antagonism. We talked about that in chapter 13. And ultimately, the body, quote unquote, trains itself to maintain a good microbiome that acts as an antagonistic effect towards a potentially bad set of microbes from coming in. And that concept is inherently more difficult to fully understand, but we've kind of touched on that a little bit here, where the body establishes a good microbiome that kind of wards off a bad set of invaders. That part is also still largely being worked out in our current state of research. An example there is in the vagina. The microbiome provides a naturally acidic environment, and it ultimately makes it difficult for outside microbes to come in. One of the problems you see with antibiotics is that it can inadvertently kill off the microbiome, and that can allow for things like yeast to overgrow, leading to things like a yeast infection. So the antagonistic effect keeps the yeast from coming in. However, in the absence of that microbiome, the yeast infection is able to take over, at least temporarily. Now, other aspects of chemical defense would be like lysosome. So lysosome is like an aspect of the physical barrier that's sort of the chemicals released from the physical barrier. So lysosome, for example, is largely found in the mouth, but also is a part of your tears. And this chemical breaks down bacterial cell walls. Lactic acid is often found on your skin, could be found in other parts of the body as well. Also makes the environment where it's found more acidic. That deters microbial growths for certain species more than others. Sweat is another aspect on the skin, kind of flushes things out, prevents an environment that's not favorable to a large number of different microbes. Hydrochloric acid in the stomach. There's actually nothing that can survive direct contact with hydrochloric acid. This is effectively battery acid, which is what you have in your stomach, ironically. The only way that microbes can make it through the stomach is if they aren't in direct contact with it. So if you eat food, for example, then they can sometimes hide inside the food. You know, there's a chunk of food and the interior of the food may not necessarily be in direct contact with the acid. Some bacteria can coat themselves with a protective layer that keeps that acid from breaking them down. So there are survival tactics, but if they're directly exposed to the hydrochloric acid, that is just overtly toxic to almost anything. So stomach acid is a very potent inhibitor, and this is one of the questionable aspects when you look at how, for example, probiotics are recommended. Things like yogurt is a good example. And theoretically, it's good to eat yogurt, it's good to take probiotics, but if they come in contact with the stomach acid, they're all just essentially eviscerated. So you need strategies when it comes to probiotics to get them through the stomach acid without being essentially liquidated. This is where good probiotics have good, thick, encapsulated pills that survive the stomach acid and only open up when they get down into the intestines past the stomach, for example. So while yogurt is good for you in theory, it's difficult to say that it's a good source of probiotic because you're just going to destroy all the bacteria in the stomach without having many of them survive. Now, when you get past the physical barriers, you start to then, if you're an invading microorganism, a pathogen, let's say you make it past the physical and chemical barriers, the next thing that you're going to deal with are a group of cells called phagocytes. Phagocytes in general have the ability to survey the body, recognize foreign matter, and then ingest and eliminate foreign matter once they recognize it as being foreign. So the process looks something like this. Surveying has to do with their presence in different body locations, but recognition comes down to that they sense a foreign portion of a bacteria. We talked about this in the previous lesson where they use pattern recognition receptors that bind to pathogen-associated molecular patterns. So they basically look for foreignness by feeling it out with receptors. If they see it as foreign, they'll then ingest it, and once ingested, they will digest it within a special kind of vesicle that breaks it all down into tiny little parts. Neutrophils are one of the most common types of phagocytes in the body. Very often they're one of the first to respond, and one of their main defenses is against bacteria. It's not that they can't do responses to other microorganisms, but that is where they tend to specialize. Monocytes are another common type of phagocyte. These are going to play a major role in the blood, and ultimately, as we'll see, they tend to develop and turn into other kinds of cells that migrate out of the blood. But they're a big role in the blood, and they often communicate with other cells if a bloodborne infection is recognized, and they can sort of communicate other cells to come in and help. They do that with the release of cytokines. Now, ultimately, where they end up playing their biggest role is they end up differentiating into another kind of macrophage, or another kind of phagocyte called a macrophage. And macrophages end up playing a much bigger role overall than monocytes do. So neutrophils and monocytes primarily are going to be found in the blood. Neutrophils can migrate out of the blood, but that's where they tend to be by default. Monocytes will ultimately leave the blood relatively early in their developmental process and migrate out of the bloodstream into various tissues. At that point, they become larger and their organelle development changes so that they're more enhanced. In other words, they become better phagocytizers. At this point, they tend to stay in a particular area, and they tend to then sort of patrol or survey that particular area. So they kind of start off as a monocyte that travels through the blood, and then they eventually move into an area and sort of stay in that area, waiting for the chance that something happens to come in. If something does come in, they have the ability to phagocytize it and potentially display that to other types of cells called lymphocytes. We'll talk more about that in the next chapter, which is a more key concept for chapter 16. However, in a more broad sense, unlike a monocyte that stays in the blood as a phagocyte in the tissue, they can not only phagocytize things more readily, but they can also signal other cells in that area. So they act as a first line of defense against invading microorganisms in that tissue, and then as a potent alarm signal for other cells to come in if they're not able to fully remove that pathogen all by themselves. So they're a more potent cell overall relative to the tissue that they migrate into. Now, another key kind of cell are called dendritic cells. Dendritic cells can also phagocytize, but their specialty is actually more as antigen-presenting cells, sometimes abbreviated as APCs. And so once again, antigen presenting is a process that activates T cells, and that becomes a mechanism for the acquired line of defense, which is really the focus of our next chapter, chapter 16. So these cells will for sure come back in that topic. But ultimately, they tend to patrol areas of the body where the external environment is, very much like the mucus lines the inner part of the body. And when we say external, we mean like cavities inside the body. So not necessarily in your skin on the outside of the skin, but more like the internal environment of your body. So it says external, but it's like the cavities where the holes of like your stomach and your respiratory tract, for example, are. That's what that means. Areas that the body comes in contact from the outside world. So they actually extend out what look like little tentacles, which I'll show you a picture here on the next slide. And those tentacles are what kind of feel out the environment. And then if they find something foreign, they swallow it. And then they migrate to the lymphoid organs, very often the lymph nodes. And in that particular environment, they'll come in contact with the lymphocytes. These would be B and T cells. So that's a whole other process for chapter 16. But these are key aspects of removing the pathogens and then, when necessary, activating the acquired line of defense. So here's a quick figure. Macrophages have, this is obviously just an illustration, but macrophages are going to look a little bit more like this. And dendritic cells look a little bit more like this. And that's what I mean by the tentacles. So these tentacles here, quote unquote, stretch out and kind of fit through different external surface layers of the cells. And they can actually grab microorganisms, swallow them, and then they're triggered to migrate away from that site to the lymph nodes. And that can activate B and T cells. We'll come back to that in the next chapter. Now, different body areas have specialized phagocytes. So in the liver, for example, you have what are called Kupffer cells, macrophages that specialize in the liver. The liver is a very potent filtering organ, pretty much your primary filtering organ, especially for the blood in particular. Kidneys as well, but I should say for the blood. So things that go through your digestive tract go through the liver first. And so this is a good way for the liver to kind of filter out pathogens. And so this is an area where Kupffer specialized phagocytes play a role in that area. So if they determine something is foreign coming through the gut, they're going to be one of the more primary early cells to remove those. In the lungs, you have what are called alveolar macrophages. These are the term is in relation to the alveoli, which are the air sacs in the lung. So here, a special kind of macrophage exists, specialized in that area, and they act as a first line of defense against airborne particles. And not only just microorganisms, but even pollutants and things like that. So as we encounter airborne pathogens, these are our first line of defense in that area. So phagocytosis ultimately is a process that is carried out by phagocytes. And a phagocyte translates to eating cell or eater of cells. So they're literally eating and swallowing cells. Ideally, this occurs against foreign cells. There are some exceptions to that, but generally speaking, you see this against foreign cells. And in order to determine that something is foreign, it has to have some kind of point of recognition. And this is where the cell has to be able to recognize it as such. And although this figure here is condensed a little bit smaller, if we look here, these bacteria have these pathogen-associated molecular patterns, PAMPs, which we talked about in the previous lesson. So these PAMPs are that's the concept of a foreign marker here. So once recognized, the phagocyte will bind to that PAMP using the appropriate pattern recognition receptor, and that sort of signals the beginning of the process, which is the attachment of the microorganism. From there, it will ingest it, so it starts to invaginate the membrane. The membrane is sort of almost a fluid, malleable kind of material. So once it binds to it, it will invaginate the membrane and pull it into a special little vesicle. And that is what's called the phagosome. It's basically a little holding cell. And then from there, another little vesicle called a lysosome will fuse with that phagosome, as you can see here, and we call that the phagolysosome. So that's phagolysosome formation. So if the phagosome is a holding cell, The lysosome is a bucket of acid. And what ends up happening is you put the bacteria in a holding cell and then you effectively dump acid all over them. That kills the bacteria, as you can imagine, slowly digests them, and then they become cellular debris, and the phagocyte then releases that debris into the body. That debris will ultimately get cleared out of the body through the lymphatic system, in theory. So the lymphatic system acts as a filtration mechanism, and that ultimately kind of gets itself out of the body through that specific mechanism. Phagocytosis can occur in a specific area of the body, or it could be something that is in response to inflammation. As the debris is cleared through the lymphatic system, this has the potential to activate additional immune cells, and if there's a sufficient amount of it, that could bring more cells into the area. That gets a little more complex outside the scope of this class, but these events can be isolated or amplified to become more of a systemic kind of events. This figure here just shows you the steps of phagocytosis. So be familiar with these general steps, but chemotaxis and ingestion relate to the recognition and the invagination of the microbe into the phagolysosome ultimately, and that leads to then destruction and then excretion. So you can look at figure 15.12 here for more details on that. Okay, so this next slide here, we're going to take a look at a little bit more detail on these pattern recognition receptors. I've talked about these several times now. So these are PRRs. These are what are found on our cells, not only on phagocytes, but also, or immune cells, but also endothelial cells. So these are cells that kind of line the body. These can also be a part of the inflammatory response, which we'll get to, but the PRRs bind foreign molecules and then induce an immune response, which could be phagocytosis or inflammation, generally speaking. There are a variety of pattern recognition receptors. TLRs are one of the most common examples of those, which we'll see those on the next slide here. Ultimately, PRRs bind to pathogen-associated molecular patterns, PAMPs, and PAMPs are things, various types of macromolecules that are exclusively unique to microorganisms. And the idea is that these are not found in humans, and therefore the body can use these patterns to identify self versus non-self. It's a little harder to say exactly how the body determines self. There is a mechanism for that, by the way, but it's a little easier without going into too much detail here to just say this is how the body determines self. The body has worked out specific patterns that are exclusively found in other creatures. And in order to then determine that they're foreign, those patterns are picked up by pattern recognition receptors. And since they only occur in other types of organisms, as soon as the body sees them, it automatically knows they're foreign. So some examples would be peptidoglycan. Peptidoglycan is a building material for bacterial cell walls, and that is literally the only place on Earth that you find it, is in the cell wall of a bacteria. Once you see that, you know you're dealing with a bacteria. Another similar example is called lipopolysaccharide, and this is abbreviated as LPS, sometimes called endotoxin, which that'll come back up here again at the end of this lecture. Also, only found here in gram-negative outer membranes, so a little more specific to the gram-negatives, but also an exclusively unique chemical. RNA in most organisms, RNA in living organisms, both prokaryotic and eukaryotic, is only single-stranded. The only time you ever see double-stranded RNA is in a specific type of RNA virus here called the dsRNA virus, which stands for double-stranded RNA viruses. So that's the only place that you find double-stranded RNA. The body immediately recognizes that as the unique aspect of a virus. Another example that's relatively common is flagellin. Flagellin is a specific type of protein found in bacterial flagella. There are many other examples. These are just some of the most common. This figure here, which comes from the web, gives you some abbreviations that show you what parts of the virus or bacteria we're looking at. And then this down here tells us what type of pattern recognition receptor it binds to. So these are specific to what are called TLRs, which are toll-like receptors, which we'll get to on the next slide here. So those are one of the most common pattern recognition receptors. So these particular foreign entities are picked up by this pattern recognition receptor, and that's sort of a more specific point-by-point reference to each foreign particle and what receptor it is recognized by. So toll-like receptors, these are the most common pattern recognition receptor, and when the body binds these, when a PAMP binds to this, it sets off a cascade of actions. So it's a non-specific defense in that it doesn't require the body to recognize anything related to a specific species of foreign microorganism. So anytime the body sees a generally foreign object, it picks up on it and recognizes it as foreign. When we look at what we refer to later as the specific line of defense, that often comes down to the difference between one species of microorganism and the next. And so the idea of being non-specific would be like differentiating between a dog and a cat, or maybe a dog and a human, maybe more broadly. The difference of being specific would be like differentiating between different breeds of a dog or different people within the human group. So like being able to see one person's face and differentiate that from another person's face. That might be what we consider specific. But non-specific would just be saying this is a dog and this is a human, and everybody in this category is a human and everybody in this category is a dog, and not really differentiating between those categories. So that's kind of what non-specific means. It's specific to the group but not to the individual. So toll-like receptors are kind of a way to feel out like what's a dog versus what's a human. And if we see it as a dog, it doesn't really matter what kind of dog it is. We just treat it like a dog and deal with it in that capacity. And as I said, in the last level of defense, what we see is ability to differentiate, so to speak. I'm speaking metaphorically here, but to differentiate between different breeds and even very specific nuances between those. So TLRs ultimately are the way that the body kind of works out self versus non-self. If we're a human, we're self. If we see human characteristics, leave it alone. If you see something like a dog, that's non-self. Deal with it. Treat that differently. And so bacteria in this analogy would be the dogs. And so once we see a dog or a bacteria, we kind of deal with those as a group in a sort of a non-specific kind of way. Okay, next here we're going to look at inflammation. Inflammation is a general way that the body responds to things like injury or infection. Inflammation is complex, and so we're going to just deal with this in a very brief way and kind of relate it to the mechanisms of innate immunity. And as it says here, in the context of innate immunity, inflammation can be a whole class and a much more broad, deep subject. So we'll keep this relatively specific. So when it comes to the inflammation as it relates to innate immunity, well, we see it as a process of infection, injury, chemical or physical, or from an inflammatory process that is brought about by various types of white blood cells or things like that. So cells in the body can sense an foreign agent or sense a physical chemical injury and then release inflammatory mediators that all kind of add to the way the inflammation is induced. So some of the cell types that induce it would be white blood cells, things like macrophages and neutrophils that we've already talked about. Those are cells that can release cytokines that act as inflammatory mediators. Endothelial cells, these are cells that line your blood vessels. And then also epithelial cells, cells that line the internal environment of your body. All of these can pick up on damage or these cytokine chemicals, and they can ultimately kind of amplify inflammation. So in some cases they will initiate inflammation, or in some cases they will sort of act in a series of steps like a cascade to amplify it. So again, inflammation in the context of the innate immune response often manifests in one of four different classical signs, very often all four together. So redness, or from the Latin word rubor, heat or calor, swelling, tumor, pain, or dolor. These are all mechanisms by which the body is ultimately increasing blood flow and increasing permeability of the blood vessels in that affected area. So what's interesting about it is that all of these symptoms manifest from sort of the same underlying cause, which is that your body is picking up on either an infection or an injury or another chemical cascade. And the result is that the blood vessels in that area dilate. And as they dilate, they become leaky, and they literally leak fluid from the vessels into the area. And as that begins to happen, the tissue swells. And the swelling is then something that can cause pain if it pushes on the nerve endings in that particular area. The redness is actually a result of red blood cells moving closer to the surface. Heat is a result of the excess fluid. Swelling is excess fluid, and pain is the nerve endings being pushed by the excess fluid. So it's all kind of a similar phenomena, which is blood components leaking out of the vessels and causing that swelling to occur. Now, functions of immunity are ultimately to immobilize and functions of inflammation, what that should say. Functions of inflammation, that's a typo there, should be there to mobilize and attract immune components to the site of the injury, number one. So basically, something's wrong, let's get the defense cells to that area. Get them in there quickly as possible. Once they get there, then those cells can be used to block microbes or destroy them so that they can keep them from spreading. And then over time, they kind of set in motion mechanisms to begin the repair process. Inflammation in and of itself is not necessarily a repair process, but it is a mechanism to begin and set in motion that repair process. It does act as a cleaning crew, so to speak, where it helps you remove harmful substances that tend to get in the way of the repair process. So it's a bit of an initiator, but not necessarily the process of healing. And in fact, that's one of the problems that you have with chronic long-term inflammation. If you're constantly inflamed and the inflammation doesn't go away, that actually impedes healing and it actually slows it down. So it's important to note that inflammation can begin and kind of get the ball rolling, so to speak, on healing, but it needs to then subside before the healing process really takes place. And so that's one of the problems with chronic inflammation. It actually acts as a deterrent to resolution. So, again, inflammation, pretty complex. These are all just basic ideas that relate to how it works with the innate immune system. Now, fever is something that can eventually happen if you have lots of these cytokines and specific types of cytokines. The body essentially can raise the temperature, and what that can ultimately do is, we'll see here on the next slide, or a couple slides here, is that it can actually begin to impede microorganisms from establishing. But fever is something that can be low grade, moderate or mid-grade fever, and a high-grade fever. And at each different level, you have different trade-offs. The body temperature goes up, it makes it harder for microorganisms to operate. It also can be more damaging to the body's nerve cells in particular overall. So different ranges of temperature have different both benefits and consequences. We don't really like to see high-grade fever, especially for long periods of time, because that can damage the body, especially do brain damage and nerve damage in particular. Interestingly, the body's temperature is a consequence of its overall level of metabolism, and that is essentially set by a part of the brain called the hypothalamus, which does other things as well. One of the main hormonal centers in the body, or in the brain, excuse me. And so the hypothalamus, in this very complex mechanism, sets this body temperature, and it also ultimately kind of does this by controlling muscle and blood vessel motion, the amount of movement muscles are conducting, and the diameter of blood vessels. And so ultimately, you can kind of change the body temperature by readjusting the musculature and the blood vessels. And so the hypothalamus kind of sets that in a very complex way. Like, goes well outside the scope of this class, but that's the kind of generality behind it there. And pyrogens are substances that have the ability to act on the hypothalamus in a way that kind of changes these settings, if you will. So if you think of the hypothalamus as a thermostat on the wall, you can think of pyrogens as a mechanism to sort of turn up the thermostat on the wall. So these can actually come from both inside or outside the body. Normally, and from a naturalistic standpoint, you have what we call endogenous pyrogens, and these originate very often from your own white blood cells. So during the context of an infection, white blood cells can release pyrogens, which are a class of cytokines, and those particular interactions of cytokines can eventually trigger the hypothalamus to raise that setting, so to speak. Cytokines are complex. They often have multiple roles, but in the context of a pyrogen, interleukin-1 and tumor necrosis factor can act as pyrogens. And where I say they're complex is they do other things as well. But they ultimately, in this context, can reset the hypothalamus to a higher level. So that's the idea of an endogenous pyrogen. Exogenous pyrogens can ultimately do the same thing, except they originate from outside the body. These are products of an infectious agent, and ultimately they kind of trick the body into raising the thermostat without it being from a natural source. These can be dangerous because they don't necessarily have the same delicacy. So as the temperature is being reset, it tends to go up more rapidly, and it's a little more dangerous to do it in that kind of way. There are a multitude of exogenous pyrogens. One of the more common types is part of the lipopolysaccharide layer that I mentioned earlier as an example of a PAMP, and these come from gram-negative bacteria. Now, not all gram-negative bacteria are made equally, so there's a little more nuance to it. But one of the subtly different components of the LPS is called endotoxin. An endotoxin, a component of lipopolysaccharide, has the ability to act as a pyrogen and cause elevated fever in extreme cases. Now, fever is a double-edged sword, as I mentioned. Depending on the grade of fever, you get sort of different trade-offs. But the benefits of fever are thought to be one of three different things. There are certain microbes that are fairly temperature sensitive. Certain viruses in particular don't do well outside of a narrow range of temperature. They're adapted to our normal body temperature, and if they go even slightly higher than that, they stop functioning. So in certain cases, raising the temperature even just a little bit can slow down or even just shut down viral replication in particular. In terms of bacteria, things like staph and strep bacteria in particular, they have to be able to absorb iron from the body. And when the temperature goes up for more complex reasons, that ability to absorb iron goes down. So temperature goes up, iron absorption goes down. And so that's one of the other reasons why it's especially effective against certain types of bacteria like strep, for example. And then in a more general sense, fever tends to speed up chemical reactions in the body. So if you raise the temperature, everything kind of gets going faster. And so this includes blood cell production, phagocytosis, the adaptive or acquired immune response. And so as everything's moving faster, in theory, that means you'll get quicker, more rapid responses. But as I said, there's a trade-off there because you can also hurt the body by increasing temperature. There's a reason that your body isn't just automatically set to 105 degrees Fahrenheit. It's not necessarily good long-term. So there's a short-term trade-off that could come at a long-term consequence. And so there's always sort of a balancing act there where what do you do with fever? Do you let it run its course? Do you try to use something to break the fever? And that's really something that a medical expert has to decide for each individual. Now, last example that we'll go through here as part of the innate immune system, I'll give you a couple of examples of antimicrobial products. So the two that we're going to talk about are called interferons and complement proteins. And so antimicrobial products are basically proteins that the body makes that sort of directly act against microbes, somewhat independent of the cells, like phagocytes. Now, they often overlap in those functions, but they technically can act independently of the cell. couple ones that are mentioned in the book that I won't go into here are antimicrobial peptides and restriction factors, or what are called host restriction factors. So I'll have you read about those last two, but we'll go through the first two here somewhat briefly. So ultimately, as kind of a general part of the innate immune system, they're there to kind of prevent microbes from establishing, protecting through neutralization and limiting their spread. But as I said, they can act directly, or they can play another sort of overlapping role with things like phagocytosis and inflammation. So they have, in a certain capacity, a way to act direct or enhance the activities of other parts of the innate immune system. So on this next slide here, we'll talk about this first one, interferons. And the term interfere on is actually the idea that they interfere with some of the activities of infectious agents. Now the book will go into more detail here. I'm going to really kind of focus on how they act against things like viruses. So interferons are complex. I guess I'll focus on their role with viruses, but they do play other roles in things like immune regulation. They can actually fight off bacteria as well. And so there are a number of different ways that these work here. In fact, there's really 3 different types of interferons: interferon alpha, interferon beta, and interferon gamma. And so these have all slightly different roles, and so we'll keep this relatively short here. It is worth noting that when you look at infection in the body, how well your body produces interferon, which is somewhat of a genetic aspect, but also an aspect of nutrition and health. If you're healthy, you tend to produce this more. If you're unhealthy, you have a harder time producing it. So interferon deficiency has sort of an indirect relation to how severely your body is affected by viruses. If you have good interferon, viruses have a harder time spreading in the body, as we'll see why here in a second. Also, interestingly, pharmaceutical companies have learned how to commercialize interferon. So they can be made in a lab and then used as a therapeutic agent to treat viruses and even things like cancers and things like that. So that goes beyond the scope of this class, but that's an interesting side note, that they're essentially a way to be used as a medical product. Now here I'm going to focus on antiviral interferons. So rather than talking about all the interferon different functions, we're going to talk about how they affect viruses. So one of the more potent known effects is that when a virus infects a host cell on the interior of the host cell, then what can happen is the host cell will turn on gene expression that now begins to change the cellular behavior in response to the virus. And it's almost like if you're at your home and an unknown person walks into your front door and you've never seen this person before and you feel like they're a threat to you, you're going to change your behavior, right? You're not just going to continue acting the same way you were 5 minutes ago. And amazingly, cells kind of have a similar ability. They actually can perceive that a virus just came into their intracellular environment, and as a result, they change their behavior. That specifically means they activate genes as a consequence of the presence of a virus. So the cell, in a certain way, knows that the virus is there, and it activates genes that are specifically targeted against viruses. So just an insight as to how sophisticated our cells actually are. Now, unfortunately for the infected cell, the interferon activation doesn't help it. So this is kind of an altruistic mechanism. What happens, though, is the infected cell, what I'm calling here the primary cell, will turn on genes that produce interferon, IFN, that is basically a signaling protein, and that protein now travels to nearby cells and binds to, this is the interferon here, travels to nearby cells and binds to specific receptors on these nearby cells. That now changes their behavior in a way that's actually different from the first cell. So the primary cell changes its behavior by releasing IFN. IFN changes the behavior by now turning on antiviral proteins or viral degrading proteins. And so it's kind of like if someone walks into your house, you don't necessarily engage them directly in this particular context. You call everybody in your neighborhood and you tell them, be on alert, there's potentially somebody that might come into your house next. Not the best analogy, but that's kind of how this works. So the cell itself responds by warning the other cells around it. So I'm calling these here the secondary cells. So the primary cell is the one that's infected. Secondary cells are the ones who are in the nearby neighborhood. So the primary cell will essentially warn the other cells. The warning causes those cells to turn on antiviral protein synthesis. And as a result, those now newly synthesized proteins will destroy the virus if and when that virus comes into their cell. So all these cells exist nearby, and as soon as the virus now exits the cell and tries to infect a new cell, this cell is now prompted to respond differently. And so that's kind of the mechanism here at play. So it makes it much harder for the virus to spread and replicate to other nearby cells as it's changing its behavior to other cells. So the idea is that you have one cell that's basically kind of giving up its ability to function by letting other cells around it know that there's an incoming virus. So that's the idea behind interferon here. Last example here is called complement. So complement is a system of proteins that ultimately allow the body to react to viruses in a very specific protein-based mechanism. So we have about 50 different proteins here, and they are constantly traveling through the bloodstream. And as soon as those proteins become activated by some kind of pathogen, they're going to change and bind to that pathogen, and that binding ultimately sets off on one of a couple different types of pathways, either the classical, the alternative, or the lectin pathway. Now these can then be further activated by antibodies as well. So the pathogen can directly activate these complement proteins, or antibodies can activate these complement proteins. And this is where I mentioned in a previous lesson about overlapping mechanisms of defense. So complement proteins ultimately set off a cascade of reactions. And those cascade reactions can lead to a variety of outcomes. Number one is that they tag the host cells, or the pathogen cells. So tagging, you can see over here in this figure, leads to what's called opsonization. And opsonization is basically putting these markers on the cell, which is flagging them for death, which is going to occur through phagocytosis. So by tagging the cells, which is called opsonization, it makes it so that phagocytes can grab a hold of them more easily. And so that sort of tags them in a direct way, but then in an indirect way makes them easier to eat by the phagocytes. In a more complex pattern, these complement proteins can form what are called a MAC attack, a membrane attack complex. The membrane attack complex ends up causing cell lysis, as you can see here in this figure. The complex actually punctures a hole in the cell, and that causes the internal contents to spew out, and that kills the microbe directly. So you kind of have an indirect mechanism in tagging and a direct mechanism in the MAC attack complex. Another more broad idea is that these complement proteins can cause inflammation. And what happens with inflammation is that the proteins basically draw in other types of cells to the environment. So these act in that kind of sense as like a cytokine, as a signaling process to other cells nearby. So that's a little more indirect here, but they kind of speed up the inflammatory process and therefore kind of draw new cells into the environment. So you can kind of see from this figure here, there's a variety of things going on. In general, they're tagging, and then sort of indirectly that causes downstream effects, which is phagocytosis, increased inflammation, cell lysis, and things like that. Ultimately, though, complement proteins are always in the blood, and they're a very rapid mechanism for dealing with microorganisms. So as soon as a microorganism comes into the body, these complement proteins can start to bind and attack, so to speak, that microorganism. Now, I have a whole video. One of the videos I have for you this week is from the video assignment. It's called Tiny Bombs in the Blood. So that video does a pretty good job of explaining these in more detail. So if you haven't watched that video, keep that in mind. this video here, or that video really talks about these more specifically and gives you a lot better visuals there for that. Last thing is just to reiterate, there are 3 complement pathways. The classical pathway, these are activated by antibodies. So the body produces antibodies through the acquired or the adaptive third line of defense, which we talk about next week in the chapter 16. So antibodies end up enhancing complement once they're produced. Lectin is a pathway activated by certain sugars. Lectins bind to sugars, I should say, and lectins then can recognize certain surface proteins and activate complement in that mechanism. And then the third pathway is activated by gram-negative bacteria, where they bind to the lipopolysaccharide, LPS. So LPS is a kind of a specific part of the gram-negative bacterial cell outer membrane, and so the alternative pathway kind of directs its activities directly towards the lipopolysaccharide layer. Okay, and we'll stop it there. And next week we'll talk about the acquired adaptive immune system, and that's your third level of defense. And we'll get into more detail and come back and readdress some of these topics to kind of remind you how they all relate together.